Hello guys, so today we're going to be talking about DNA replication. This is just a simple drawing to show you what the end product is going to be after this process. So we have DNA, and DNA replication, like we talked about in class, happens during the S phase of the cell cycle, and it happens inside of the nucleus. DNA replication, all that is just is a process where DNA makes copies of itself. Okay, so this is a very simple representation of that, and as you can see, we have a DNA molecule there, double helix, anti-parallel, and the first thing that needs to happen, or the first step, is going to be the unzipping of this of the DNA strands. So we separate the strands, and the enzyme that does that is DNA helicase. So it unzips the DNA, and then DNA polymerase comes in, and it actually adds complementary strands to those to the parent strands to make two identical DNA molecules. Okay, and that's the entire process. Okay, very simple way of explaining it. We go from one DNA molecule to two DNA molecules that are identical to the parent DNA molecule. Okay, and that's important because when your cells, when your cells are dividing. Okay, especially let's think about your skin cells. When your skin cells divide, then you want those cells to have the same DNA inside of them so that they can produce the same proteins, and you can have you can replace the skin that you had, okay, with the same identical cells that you had. Okay, an example of this, we can write it. We can actually use the nitrogen bases or the letters ATCG, so that's representing the DNA. So the first thing that would happen is that strand would actually split, just like in this example, and then DNA polymerase would add those complementary strands. And if you look at those strands that are in the boxes, you'll see that they're identical to the parental strand, or the first one. Okay, so again, this is just DNA replication. Okay, the method is called semi-conservative method because you always keep half of the original strand when the DNA is replicating. Okay, and this is just a very simple way of showing you what replication is, which is just DNA making copies of itself. Okay, so now I'm going to show you a different drawing where I'm going to go into more detail about the process. Okay, these lines right here represent a DNA molecule, and you can see I'm going to add some nitrogen bases, ATC, ATC, and the complementary one is TAG, because we know by the base pairing rules that A always bonds with T, T always bonds with A, G always bonds with C, and vice versa, C with G. Okay, now when your cells are reading the DNA, okay, and when DNA is replicating, DNA is read in sections of 3 nitrogen bases. Okay, and that section, or those 3 nitrogen bases, are called a codon. Okay, and that's going to be important, especially when we get to protein synthesis, when we start talking about anticodons and how DNA actually codes for proteins. Okay, so those those sections, the section of 3 letters or nitrogen bases represent a codon. Okay, the process, remember, that happens in the nucleus. We know that DNA is a double helix and is made up of nucleotides. We said nucleotides are made up of 3 parts, which are the phosphate, the sugar, which is deoxyribose, and one of the nitrogen bases. Okay. Now those nucleotides are bonded together by hydrogen bonds. Okay, they're very weak, and that's important. That's important for replication of DNA because remember, like we said in the previous drawing, DNA has to unzip. So you want these bonds to be weak so that when it's time to unzip them, it doesn't take a lot of energy to separate the strands. Okay, DNA is also anti-parallel, meaning that it grows in different directions. Okay, parallel lines never touch each other. As you can see, DNA never touches each other. It looks like it does in this drawing, but you know it's just twisted around. It's a double helix. And it grows in opposite directions. The 5' and the 3', we'll get to, we'll talk more about that. But all they represent is the carbon number to which phosphate is attached to in the glucose molecule. Okay, we know that the backbone of DNA is made up of phosphate and glucose, and all those numbers represent is that the number of the carbon that the phosphate attaches to. Okay, And again, it allows us to see and how DNA is anti-parallel. Okay, never touches, the strands never touch each other. They're parallel to each other, but since they're growing in different directions, then they're called anti-parallel. Okay, so the first thing we're going to add is the DNA helicase. The green structure there represents the DNA helicase, which is the enzyme that's responsible for splitting the DNA molecule. Okay, and the location where the DNA molecule is split by the DNA helicase is called a replication fork. That's all that is. It's just the location where the separation or the unzipping of DNA happens. Okay, the enzyme attaches, separates the strands, creating a replication fork. Then the other important enzyme during DNA replication attaches to the daughter strands, okay, the strands that are separated, and it's called DNA polymerase. And what it's going to do is it's going to start reading the nitrogen bases that are in the original strand, and it's going to start adding them from a 3 to 5 direction, okay, and it's going to start adding the complementary strand. And you can see that C always wants with G, G with C, T with A, and A with T. Okay, and what's now in the other strand, you see that I put the nitrogen bases as well. And the difference here, or what I want you to pay attention to, is this gap right here. Okay, there's a gap right there, and that's different than this strand. This strand has no gaps. Okay, this one is continuous. There's no breaks in this one. There's a break right here, and because of that, this one is going to be called the leading strand, or it's called the leading strand because it grows continuously. It doesn't stop. Okay, it grows much faster than this one, and the reason for that is because it's growing continuously. The other one, the one that grows in fragments, is called a lagging strand because it's lagging a little bit behind than the leading strand. Okay, and it's made in fragments. Those fragments are called Okazaki fragments. Okay, and they're going to create this little gap right there. Okay, and since they have a gap and they're made in fragments, well, they're going to require an extra step for that gap to be filled up, and that's where DNA, this yellow structure right here, DNA ligase, shows up. Okay, and what it does is going to use an enzyme that's going to actually bond these strands together. Okay, and it puts the fragments together, and now we have a DNA strand that has no gaps. Okay, now the original strands, like we said earlier in the simpler structure, is called a parental strand. Okay, so that's the parental strand, this one right here. Okay, that's a parental strand. And these other ones, the new ones that are being made, they're called daughter strands. Okay, so the new strands, the strands that are being copied, they're called daughter strands. Okay, and this entire process, like I've been saying in the video, happens inside the nucleus, and it happens during the S phase of the cell cycle. Again, that's replication. Replication is just the process of DNA making copies of itself, and 3 enzymes are involved in them. One, DNA helicase, which splits the DNA molecule. DNA polymerase, which adds the complementary nucleotides to the DNA, to the parent strand. And DNA ligase, which is responsible for putting together the Okazaki fragments in the lagging strand. Okay, make sure you review your notes. Watch the video again if you need more clarification, and you're always welcome to come to tutoring if you want me to go over some specific questions that you have. All right?